Hello, I'm Kyung Kim from Yonsei University. Today I'll present MUST, or Cvpr 2026 paper on multimodal cancer survival prediction. MUST tackles a problem that breaks most multimodal models in practice, the missing modality problem, and solves it through a principled idea. Decompose what we have, then generate only what is truly missing. Here is the central insight. When one modality in genomics is missing at test time, can we just recover it from the modality we still have, like histology? The answer is only partially. The modality carries two kinds of information, a shared component C, which is determined by the other modality, and a modality-specific component U, which is not. The shared component is deterministically recoverable. MUST has three components. First, a low-length subspace that algebraically separates shared and specific information. Second, a latent diffusion model that generates the missing specific components conditioned on the shared part. Third, an inference procedure that activates on only where a modality is missing. Training uses complete modalities. Missing modality handling lives only at inference. The decomposition is algebraic, not learned by a separate network. We project this modality's representation onto a learned low-rank shared subspace using projector p-cap. The projector c-hat is a shared part. The orthogonal residual u-hat is the modality-specific part. Three constraints make this decomposition meaningful. Shared consistency. The shared parts from both directions must agree. Intermodal orthogonality. The two modality-specific residuals must not overlap. Intramodal orthogonality. Within each modality, shared and specific parts must be disentangled. Together, these turn missing modality recovery into a well-posed problem. So, why diffusion? Recovering the missing specific component means modeling a multimodal conditional distribution, not a single point. An MLP corrupts to deterministic mappings and cannot represent multiple valid realizations. A VAE suffers from latent entanglement and mode averaging, so its samples lose modality-specific tail. A conditional latent diffusion model handles this naturally, generating diverse, plausible samples consistent with the available modality. Inference is a clean four-step pipeline. Say genomics is missing. We only have GP from pathology. Step 1. Low-rank projection extracts the pathology-specific part U hat P. Step 2. Decomposition isolates the shared part C hat G from P. Step 3. Shared consistency lets us treat that as the conditioner for genomics. Step 4. Conditional latent diffusion generates the missing genomic specific component. For stability, we sample 5 times with DDIM and average. The result is a complete representation, recovered, not hallucinated. We evaluate on 5 TCGA cohorts: BLCA, BRCA, GBML-GG, IUAD, and UCC. Using paired whole slide images in genomics, genomic features are organized into 6 pathway groups grounded in cancer hallmarks. WSIs are encoded with a pre-trained patchwise pathology encoder. We use Herald's C-index with 5-fold cross-validation, reporting mean and standard deviation. First, we complete modalities must achieve the best C-index on all 5 codes and an overall 0.742. The comparison with CMTA is particularly informative. Both models use bidirectional cross-validation but must improve from 0.724 to 0.742. Since the fusion mechanism is the same, the gain comes from the decomposition structure itself. Disentangling shared and specific information produces a better representation even before missing modalities enter the picture.